Modern microscopes used for biological imaging are expensive, are located in specialized laboratories and require highly qualified staff. To research novel, creative approaches to address urgent scientific issues, for example in the fight against infectious diseases such as COVID-19, is thus primarily reserved for scientists at well-equipped research institutions in rich countries. A young research team from the Leibniz Institute of Photonic Technology, Leibniz IPHT, in Jena, the Friedrich Schiller University and Jena University Hospital modular system can be combined specifically in the way the research question requires, from long-term observation of living organisms in the incubator to a toolbox for optics education. The basic building block of the UC2 system is a simple 3D printable cube with an edge length of 5 cm, which can host a variety of components such as lenses, LEDs or cameras. Several such cubes are plugged on a magnetic raster base plate. Cleverly arranged, the modules thus result in a powerful optical instrument. An optical concept according to which focal planes of adjacent lenses coincide is the basis for most of the complex optical setups such as modern microscopes. In this way, UC2 also provides users without technical training with an optical tool that they can use, modify and expand, depending on what they are researching. The UC2 system allows us to produce a high-quality microscope at low cost, with which we can observe living cells in an incubator, he states. UC2 thus opens up areas of application for biomedical research for which conventional microscopes are not suitable. For a study at Jena University Hospital, the UC2 team observed the differentiation of monocytes into macrophages in the incubator over a period of 1 week in order to gain insights into how the innate immune system fights off pathogens in the body. Building according to the Lego principle. From the idea to the prototype building according to the Lego principle, this not only awakens the user's inner play instinct, observes the use. If an inverted microscope is needed, you stack the cubes accordingly. With the UC2 system, elements can be combined depending on the required resolution, stability, duration or microscopy method and tested directly in the rapid prototyping process. The vision, open science. The researchers publish construction plans and software on the freely accessible online repository GitHub, so that the open source community worldwide can access, rebuild, modify and expand the presented systems. Thanks to the detailed documentation, researchers can reproduce and further develop experiments anywhere in the world. UC2 Experiment Box brings science to schools in order to get especially young people interested in optics. The research team has developed a sophisticated tool set for educational purposes in schools and universities. In Jena, the young researchers have already used the UC2 toolbox in several schools and E.g., supported pupils to build a fluorescence microscope to detect microplastics. This enabled us to build our own fluorescence microscope cost-effectively without any major optical knowledge and to develop a comparably simple method for detecting plastic particles in cosmetics, reports Amelia Walter from the Montessori School in Jena, who together with her group is pursuing. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.